It really gives me a great pleasure to introduce my good friend Pak-Wing Fok from the University of Delaware. Now, he has a really interesting biography. His family's from Hong Kong, but he was born in Singapore, grew up in the United Kingdom. You'll recognize that accent instantaneously. In most of his education in the United Kingdom, but in continuing traveling around the entire world, he came to MIT to get his PhD in mathematics, and then did postdocs. I got this right, UCLA and Caltech. Wow, I got the whole thing memorized. And he's been working on models, mathematical models in atherosclerosis for a number of years. And I really think he's thought more about this and better about this than anybody else I know. So we're going to hear about it right now. Thanks so much.
Okay, hopefully that's good. So thanks, Bill, for that wonderful introduction. I've never given a talk at a hospital before, so this is my first time. So I'm a mathematician, and I'll tell you more about my background in a minute. But I've been thinking about atherosclerosis for the last 5 or 6 years, and trying to learn more about the clinical side, biological side of this very interesting but complicated. Okay, so as Bill said, I'm a mathematician. My PhD was nothing to do with biology or medicine. It was actually material science. But when I went to UCLA, I was in the biomathematics department. That's where I started to get interested in biology. And then when I came to Delaware, Bill actually introduced me to this problem of vulnerable plaque. And I've been working on it since then. I think it's a very fascinating question. Tried to write a few papers on it. And the work that I'm presenting today is kind of my latest, my latest set of results. So I hope you find it interesting. Okay, so for today, I'm going to talk about a bit about the pathophysiology of atherosclerosis, and then this phenomenon called Glagov remodeling, which I find to be very fascinating. I'm going to assume that most of you have maybe some familiarity with mathematical models, but maybe not that much. Maybe you're rusty with it. And so I will tell you a little bit about mathematical models generally, and then I'll concentrate on a mathematical model of intimal thickening. And this model is motivated mainly from animal experiments, and it comes from an area of mathematics called hyperelasticity. I'll be comparing my model to data, and then maybe there's a possible medical utility. That's purely hypothetical at this point, but I guess you guys can give me more input on whether it might actually be something that could be realized or not. Okay, so atherosclerosis. What is atherosclerosis about? So atherosclerosis is mainly a disease of the intima. The blood vessel consists of 3 layers. There's the intima, and then there's the media outside of that, and then the adventitia, which is outside of the media. And atherosclerosis is mainly a disease of the intima, and it results in growth and changes in the intima. So, for example, you can get buildup of cells, dead matter, extracellular matrix, calcium phosphate. All these things give rise to something called plaque. And there are many different types of plaque and different stages of atherosclerosis. This is a bit of a small picture here, but these are IVUS scans, right, ultrasound scans of different types of plaque. And each plaque has a different kind of abbreviation. So AIT stands for adaptive intimal thickening. PIT stands for pathological intimal thickening, and so on. So each plaque has different characteristics. In particular, there are some plaques that are very, very dangerous. These are plaques that have necrotic cores. So you can see in these pictures here, NC stands for necrotic core. This is a vessel cross-section. And these plaques are highly eccentric. You can see the buildup of the dead matter in the cells here. And then the NC, that stands for necrotic core. On the left-hand side, or on your right-hand side, there's a significant calcification in the plaque. So these two are very advanced stage plaques. And a lot of people would say that the sign of foam cells is the first sign of atherosclerosis. Foam cells, they are macrophages that have engorged too much lipid, and so they become these foam cell phenotype. Atherosclerosis is a very complicated disease, so I'm going to do what mathematicians often do, which is just to kind of step back a little bit and try to think of a slightly more simple situation. And that's just kind of remodeling and how the vessel changes shape as the disease might build up. So again, on your left, you have vessel cross-section with the 3 different layers. And then on the right is a picture I took from this paper by Mulvaney, which shows you the different ways that a vessel could remodel. So if you focus on the middle one, that's kind of like a regular vessel. If there's a buildup of mass inside that vessel, well, actually, there are many different outcomes that could occur. So look at the hypertrophic case. So if you look at the lumen could shrink, could stay the same size, or it could expand. And so there are actually many different outcomes, even though there's a buildup in mass. It's not clear for these cases you're going to get. And that's kind of something I'd like you to keep at the back of your mind throughout this talk. Okay, so now the main thing that I'll talk about today is this phenomenon called Glagov remodeling. And look, Glagov was a doctor, I think, at the University of Chicago, and he wrote this paper in 1987. It was published in the New England Journal of Medicine, so a very famous journal. And he was the first to observe this very, very interesting vessel shape change as it develops atherosclerosis. And on the chart on the left is the data from his paper. Right? So what we're seeing here on the y-axis is the lumen area, and on the x-axis is the stenosis. Right? The stenosis is a measure of how stenosed the vessel is. 100% being completely occluded, 0% being not occluded at all. And lumen area is just it's the area of the lumen, right? In this case, represented by the circle. And his main finding from this data was that the lumen area would, in the beginning, increase a little bit before rapidly decreasing as the disease got more severe, right? So I think of stenosis as being kind of like time, because as time goes on, the vessel becomes more and more unhealthy. And so as time goes on, the lumen area doesn't shrink straight away. In fact, it gets a little bit bigger. That's called compensating or compensation before constricting, right? And this diagram here, I drew it, but actually it's adapted from Glagov's paper, right? So he has a very, very similar paper, very similar diagram in his paper. So what happens if you kind of look at the dotted lines that trace the lumen, right? There's a slight expansion, or maybe, slight expansion until you get a critical stenosis of about 40%, and then there's a rapid deterioration, and your vessel gets more stenosed in that case. So that's Glagov remodeling, and it's something that I want to try to explain using mathematics. So Glagov remodeling has been talked about by other people. So actually, just fairly recently, last year, there was a paper written by these people. And so they were talking about Glagov remodeling from the prospect study. So the prospect study is quite different from the Glagov data, because Glagov, he considered human hearts, and people who died and hearts. In this prospect study, I believe that the images were collected using IVUS, so they were in vivo. But they saw the Glagov phenomenon, the important thing. They did observe this interesting way that the vessel changed shape as it got more diseased. And then Michael Jonah and Renu Vermani wrote an editorial on that, commenting on the significance of this paper, and they were saying that Glagov remodeling is very complicated. It involves a lot of genetic factors and so on. The MMPs are important, necrotic cores are important, a lot of complicated things. And that kind of seems to be the trend in explaining the Glagov phenomenon. It's very complicated. So, for example, in this paper by Korshunov, again, they looked at mice, and they said it's due to genes involved in selectin and fermentin and nitric oxide. Complicated bag of genetic factors, and somehow they all contributed to Glagov remodeling. So I want to propose that actually Glagov remodeling is very simple. It's not complicated at all. And so this is what I'm going to tell you about today. Okay, so our current understanding of remodeling is qualitative and somewhat descriptive. So these are some of the sentences that I took from the previous papers. So they say necrotic core enlargement and the presence of calcification have been shown to correlate with positive remodeling. The occurrence of luminal narrowing seems to be a consequence of changes in vessel wall composition, especially related to the presence of macrophages. So these are all nice, but why is it that positive remodeling occurs before negative remodeling? Why does the vessel expand first before contracting? I mean, why doesn't it contract first and then expand, for example, right? So, I mean, these observations don't really explain the ordering of the remodeling, right? So here's where I think that mathematical modeling can really play a useful role. And the model is kind of motivated by some fairly old experiments done in the 80s on rats. So in these experiments, they're called balloon catheter injury experiments. They take some rats and they take a catheter and then they denude the endothelium by rubbing the endothelial cells away. It causes an injury in the vessel, the release of growth factors, and subsequent thickening of the intima. And then they sacrifice rats regularly, and they measure the thickness of the intima. And the sequence of images is kind of what you see on the left-hand side here, right? So there's injury. So 2 days after injury, 7 days after injury, 2 weeks after injury, 12 weeks after injury, you see a gradual buildup of the intima, right? So there's a thickening of that layer. Intimal thickening, I think, clinically is not the same as atherosclerosis, but it is related to atherosclerosis. And Schwartz, in his paper, he says that the atherosclerosis may begin in the soil of the intima. So it's kind of a place where the disease can develop and grow. Okay. And intimal thickening is also present in graft vascular disease. Again, it has some pathological similarities with atherosclerosis. And if you look at the GVD in monkey aorta, the lesions are pretty concentric. They're not eccentric like the human lesions that I showed you in the beginning. The main difference with GVD is that it can develop very, very quickly. Right? So my mathematical model is kind of inspired by GVD and by the animal experiments. Okay, so now we're going to get to the slightly more mathematical part of the talk. And this slide is intended just to give you an introduction to mathematical models. So take a system that we're all familiar with, the weather, right? So we can make predictions about the weather. How do we do that? Well, there are two components. There are two pieces. There are the equations, and then there are model parameters. Right? So for example, we've got to take current weather conditions, we've got terrain information, the amount of solar radiation that falls into the planet, the melting point of ice. These are all fixed parameters of the model. Right? You also have the governing equations. The governing equations consist of fluid mechanics, Navier-Stokes equations, geostrophic approximations, cloud physics, thermodynamics, and so on and so on. Right? And these are all put into a computer, and a very, very big computer, and the computer will spit out things like temperature prediction 3 days from now, 5 days from now, 7 days from now, precipitation levels, wind speeds. Right? I mean, you're all used to this. You're used to mathematical models playing an important role in this, right? And we can reap a lot of benefits from mathematical modeling in terms of agriculture and hurricane forecasts. So this is very, very valuable. But what we really need, I mean, the people who do these, solve these equations, they really need input from meteorologists and weather experts, right? You cannot underestimate their role in this. So it's really important that the meteorologists communicate with the engineers and the mathematicians behind these models so that they can come up with meteorologically realistic set of equations. And you can argue exactly the same thing about atherosclerosis, right, and intimal thickening. So again, there are some governing equations. And those governing equations, I have a feeling for what they could be, but I need help from clinicians and imaging experts to come up with the governing equations. Right? But very loosely, those equations would come from Newton's laws, for example, the laws of physics, nonlinear elasticity theory, theories of tissue growth, diffusion of growth factors, and there will be model parameters like the blood pressure, vessel layer properties, intimal growth rate, dimensions of an unloaded artery. These would all be fixed parameters or constants in the model, right? And again, you put all these equations into a computer, you run your program, and potentially it could spit out a lot of valuable information, right? Like how your vessel dimension would change over time and how the stresses change over time, right? And in the same way that weather prediction becomes very useful, in the same way that meteorology becomes very useful once you can predict in the future, same goes for this, right? I mean, imagine if you could predict what happens to a patient in terms of them, how thick their intima is in 5 years from now. Okay, the next few slides are the mathematical theory of my model. So kind of underpinning my project is this theory called Morphol Elasticity, which is a theory of tissue growth. And this is actually a fairly old theory. I didn't invent it. You can find out more details in these two papers. But essentially, it takes an unstressed reference configuration, and it says that, well, your current configuration is obtained by going through two separate steps. There's a growth step followed by an elastic response, right? And so the Morphol Elasticity assumption says that the deformation gradient can be decomposed into an elastic tensor times a growth tensor, right? Kind of that's very mathematical, I realize, but in kind of in common sense terms, imagine you had, say, a rod wedged between two immovable walls, right? And that rod was to grow a little bit. That would be the growth step, right? But because the rod is trapped between these two walls, there will be an induced elastic response, right? It would grow, but there will be stresses that develop as a result of that growth because it's stuck between these two walls. And so that would be the elastic response, right? So that's basically what Morphol elasticity is, but done now in 2 dimensions and 3 dimensions. Okay, so in the mathematical model, I'm going to assume that we have concentric lesions, right? So I'm just going to assume that the intima, the media, and the adventitia are all annuli. So they're just rings, right? And what I'm basically trying to keep track of is the mapping from the unstressed reference state to the grown pressurized state. So at time t is equal to 0, I assume that there's a blood pressure that is put in, that you load the body with the blood pressure. Right? So this is going to cause the whole system to dilate a little bit. And then for T positive, I'm going to say, well, the intima is going to grow. Right? You can see that actually the pink region here is a little bit thicker than the pink region here. So the intima has grown. What is the response of the artery to this growth? Right? And so this is some of the mathematics that underlies that. The elastic response is F sub E. F sub G is the growth tensor. The growth tensor is diagonal. So it takes the form diagonal GG1, and G is the growth here. Right? The map from A to C is found by solving this ordinary differential equation 5. And there's an initial condition that says that I'm in this state in the beginning. Right? And so for more details, we have a paper that's under review for mathematical medicine and biology. There's also a copy on my website if you want. So our model also contains elements of stresses and strains. So these equations tell you that essentially you've got a blood pressure P inside the lumen, and that the stress has to be continuous across all the interfaces. This part here is to do with mathematical modeling of the mechanical properties of the individual layers. Okay? So imagine that your vessel is consisting of 3 different layers. Each layer is going to have different mechanical properties. They're going to respond differently to forces, right? And the way that we can prescribe the mechanical properties in the context of nonlinear elasticity theory is by prescribing something called a strain energy density function, which is this W here, right? So this looks very complicated, but just think that the W gives you all the mechanical information you need about the vessel layer, about how it deforms and how it responds to stresses. Right? That's all encapsulated in the W. It's like it's a fancy version of Hooke's law, if you like. Now this W function has a lot of unknown parameters: the mu, the eta, the beta, the rho. Right? All of these can be found from doing loading experiments on these vessels. Okay? And I'm going to show that to you now. So there have been people who spend their entire lives performing mechanical testing of arteries. Right? So these kinds of graphs is what they typically get, right? So they apply certain forces onto the arteries and see how much they extend by. And by looking at their data and by fitting the red curves, they're able to infer the parameters of the strain energy density function, right? They're able to infer these constants, the C, the K, the K2, and the beta, right? So the mechanical properties of the arteries, they do come from performing actual experiments. They're not made up by me. One thing that is good to note here is that the media and the adventitia have two sets of collagen fibers that kind of wrap around the vessel wall, right? So they form an angle 2 beta M here, 2 beta A here. That's not that important here, but the presence of these collagen fibers are important, and I'm going to talk about them later on. These collagen fibers become very, very stiff after you extend them, right? And that's going to be quite significant, actually, when it comes to explaining Glagov remodeling. Okay, so here are my results. So I took all that mathematics that I showed you, I put them into a computer. There are a lot of equations that you've got to solve, so you run them. I spend a lot of time thinking about how to solve them the best way, and so on. And this is what I get in the end. So the red dots are data from, in this case, rabbit experiments, right? So they took some rabbits, and again, they sacrificed them regularly, and they measured their intimal area after denuding the endothelium. And I'm able to get my model to fit the data by choosing the correct parameters, right? The media does not change generally, and that's also a consequence of my model. I assume that all the growth happens in the intima, so the media does not change area at all. The bar graphs at the bottom here, they're just the results of the fitting procedure. I used a procedure called the Metropolis-Hastings algorithm to try to find the best fit curve, which is a solid line, along with 95% confidence intervals, which are given by the dashed curve. I can do the same thing with rat data. But again, this is from the old study in the 80s. There are fewer data points here in this case, but again, I'm able to perform the same procedure and fit my model in the black line to the red data points, right? And so what this model is really illustrating is the thickening of the intima that we see in A, B, C, D, E. Okay, so that's fine and well. You can say, well, I've taken my model and I've chosen the parameters to fit the data, right? What can we learn from that? So notice that in these graphs, I'm only showing you the intimal area. Right? I'm not showing you anything else. I don't know anything about the lumen. I don't know anything about the media or the adventitia. Right? But I can fit the lumen. I can fit the intima area. The intima area is the only data that's published. Right? They don't publish details about the other vessel layers. Right? But having fitted this data, I can now say quite a lot of things about all the other layers. Right? So this is a much more detailed trajectory of the evolution. Right? So here, I'm plotting A, B, C, and D. What are A, B, C, and D? They're the radii of each of the different layers. Right? So A, for example, is the radius of the lumen. Right? B would be the radius of the internal elastic lamina. So on the left, it's rabbits. On the right, it's rats. And in each case, we're getting a growth. Right? So this part here is actually the intima, right? Because it's just the region between A and B. Right? Here, this is the intima. It's the region between A and B. For every time t, right, my code or my simulation gives you values of A of t, B of t, C of t, and D of t. And that completely describes the dimensions of your vessel. Right? So potentially it gives you a lot of information. And interestingly enough, this is saying that in the case of the rabbits, in the beginning, the vessel is compensating. Right? You don't see very much change in the lumen area. Right? The lumen area would just be pi times a squared in this case. Right? It's just a circle, pi a squared. So there's no change in the lumen area. But as there's more and more growth, then what happens is that the media and the adventitia, they yield. Right? They're compliant and they yield. And this is the kind of growth that you get. So there's compensation and then outward remodeling. In the case of the rats, we actually see either compensation or a little bit of increase in the lumen area before a decrease in the lumen, right? This is the Glagov remodeling, right? So my model predicts that Glagov remodeling does occur in the rat carotid artery, even though Glagov was not mentioned at all in their paper. And the nice thing about having this code, this simulation, is that I'm able to very, very precisely control some of the parameters in the model. For example, what I can do is I can change the material properties of each of the layers. I can change the intima stiffness, for example, right? So going from A to B to C to D, I can change the stiffness and observe what happens to the vessel remodeling, right? So again, here, I'm plotting the radii as a function of time, right? So for each time, my code outputs the radii of each of the layers, A, B, C, and D, right? And the general trend is that initially, there is a compensating stage, right? The lumen doesn't change its area very much, but instead, the adventitia and the media are yielding and expanding, right? But then you see something very interesting that happens. You see here, the adventitia and the media, they're not changing at all. They've become very, very stiff because of those collagen fibers. And the collagen fibers have been fully extended, and that makes the outer layers of the vessel extremely stiff. Almost, you can think of it like being like steel, for example. It doesn't give at all. And the intima, in that case, has only one option, and it's got to yield itself. So when it grows, the only thing it can do is to grow inwards, right? And you see the drop in the lumen area in that case, right? You see, once we get to here, no matter how much growth there is, there's basically no yield in the outer two layers, right? And when we reach this critical point, that's when you get a sudden drop-off in the lumen area. Okay? So what I'm trying to tell you guys is that I think the Glagov phenomenon is related to stiffness in the vessel wall, right? Yes, it could be explained in a very complicated way through genes and nitric oxide and lectin and necrotic cause, but it could also be explained just by thinking about the stiffnesses of each of the layers, right? When you increase the intima stiffness in my simulations, go from A to B to C to D, what I see is that the expansion of the lumen, that stage, the compensation stage, that becomes more pronounced, right? So here it's flat, but then as we increase the intima stiffness, it goes up instead of becoming flat, right? And that kind of makes sense because the intima is so stiff, it's able to push out against the other layers, right, as it becomes stiffer. And the insets, by the way, they're supposed to be my mathematical predictions of the initial Glagov data. If I can just go back to one of the very, very early slides here, right? So you notice it's the lumen area as a function of stenosis. The dots are the data. The red here, the red curve is just a best fit curve. But I can actually generate these curves from my model, right? From a model of the artery. I can generate predictions of the lumen area as a function of the stenosis. That's exactly what I have here, right? Lumen area as a function of the stenosis, right? So flat, compensating, and then failure, right? Here it's compensating, and then you get the encroachment as the stenosis increases. And also, it's 40%. See? 40%. Just like what Glagov predicted. Okay, so I guess one of the main conclusions of this study is that I've come up with a very intuitive explanation for Glagov remodeling, and the explanation is very, very simple. I think everybody in this room can understand that, right? It consists of 4 steps. Initially, the intima is the stiffest layer, right? The media and the adventitia are relatively compliant. So when you get growth, what happens? The media and the adventitia, they have to yield, right? So that's why you get outward remodeling. But after a critical stage, when the collagen fibers have extended to their maximum capacity within the outer 2 layers, the adventitia and the media, they become very, very stiff. They become the stiffest parts of the blood vessel. And what does the intima do? The intima can only contract as a response to that, right? So as a result, the intima has to yield as a result of that, right? So it's very simple, right? I think everybody in this room can understand that. It's just to do with a competition between the stiffnesses of the different layers. That's why you get Glagov remodeling. That's what I think. I don't know whether it's true. I mean, this has to be verified, but that's. I think I've played my role as a mathematician here to generate interesting hypotheses that can be tested in the lab. Okay, I guess I should talk a little bit about the genes though, because this is something that a lot of people have worked on. So there is evidence for the Glagov remodeling being associated with certain genes and proteins. I mentioned nitric oxide, vimentin, p-selectin, MMP9, and so on. Right? So these processes are important for Glagov remodeling. But the question is, do they cause a switch in the remodeling? Right? Does it cause the switch from going outwards to inwards? If they do, then these genes must somehow be able to sense stenosis fraction. They must be able to sense kind of global information about the vessel wall. Right? How would they do this? I'm not aware of any mechanisms. But maybe there is a way. So my model predicts that the Glagov remodeling is actually just an intrinsic property of the vessel itself. It's not really that complicated. It's just a feature of nonlinear mechanics, actually. It arises simply because the vessels are elastic and respond in different ways to growth, and they become stiffer as they grow. Right, so this is a very simple Occam's razor type argument for Glagov remodeling. As a mathematician, it's my duty to go for the simplest possible theory when presented with two theories that have the same explanatory power. Okay, so this is a very speculative slide, and I'd be happy to entertain questions about this later on. But I guess this is a more medical, has more medical utility perhaps. And we use angiography to detect outwardly remodeled plaques. So the problem with initial stages of Glagov remodeling is that the lumen stays roughly the same shape. So when you do these angiograms, the angiograms only highlight the blood basically, right? So you only see the lumen. You don't see the other vessel walls. So here, in this case, this was a plaque where there was significant constriction. So the plaque has narrowed here, right? But if I show you this, I think it would be quite hard to discern any outwardly remodeled plaques. Right? You can't see it because you can only see the lumen. So how can my work help you identify outwardly remodeled plaques? Well, this is just a hypothesis. But take a healthy vessel, take a diseased vessel. Right? So healthy vessel, this is time. This is the wall radii. These are the graphs that I've been showing you the whole last 40 minutes. So this is a healthy vessel. This is a diseased vessel. The diseased vessel is growing. You can see the thickening of the intima here. Here the plaque is asymptomatic. Right? The lumen is not changing size, and it's very, very hard to detect through an angiogram. But the interesting thing is that even though the plaque is asymptomatic at this stage, it has different material properties. In fact, it's harder. It's a stiffer plaque. Right? And what that means is that it's going to respond very differently to something like a vasodilator. Right? So if you apply a vasodilator to both the healthy and the diseased vessel, they're going to respond quite differently. And according to my calculations, there's going to be a much bigger change in the lumen area for a healthy vessel compared to a diseased vessel. Because the diseased vessel, the lumen area only changes by a little bit because it's so stiff. Right? Whereas a healthy vessel is very compliant. Right? So it'll change by a lot if you apply like ACE inhibitors or a diuretic, something like that, right? So you can see that I simulated, a course of vasodilators for a short amount of time. In the middle is a big yield healthy vessel, right? But only a very, very small yield in the diseased vessel. And the yield is in the lumen. So that can be detected. It could potentially feature in the angiogram. So that's my harebrained idea. Who knows whether it's actually going to work or not, but that was a thought that I had. Okay, so talk about the next step. So, I'd like to relax the assumption of asymmetry. At the moment, I'm dealing with this concentric annuli as my geometry. Real plaques obviously are not perfect circles. They tend to be eccentric, so that should be incorporated. I could also potentially include molecular genetic factors and create so-called virtual plaque. That might be a very useful tool to the community. And also, I'd like to further develop collaboration with imaging experts and medical professionals such as yourselves, which help me refine my model and make it a lot better. Okay, so this is my final slide. I guess I'm going to end a little bit early, but my takeaway messages are these. So Glagov remodeling is biologically significant because it arises in both humans and in animals. Right? to date, there hasn't been a satisfactory explanation for this process. Using theory from nonlinear elasticity theory and tissue growth, I found out that Glagov remodeling arises essentially due to the outer layers becoming stiffer as the disease burden increases. Right? But this hypothesis still needs to be validated by experiments, and the model suggests the possibility of detecting hard plaques through angiography. If you'd like more details, as I said, we have a paper under review for mathematical medicine and biology that's available on my website. I'd like to thank my collaborator, Rebecca Zanft, who has been instrumental in helping me. She's actually the mechanics expert in this. And also, I'd like to acknowledge the support of the Simons Foundation, who's given me a small grant to go and see Rebecca to further our collaboration. So I'm aware that I'm a little bit under time, but that just means that we have more questions, right? So thanks a lot for your attention. Thank you.
Well, it was fabulous. I learned a ton. That people looking for genetic causes of Glagov remodeling reminds me of the drunk looking for his keys under the street light, because the people that are doing that's what they think about.
Yeah, right.
But sometimes a mechanical explanation like this is so much simpler and so much more compelling.
And it's intuitive, right? And
very
you can get it, you get it.
It's very easy.
Yeah.
Without understanding any of the mathematics, you can understand exactly what you're talking about.
Right. So I think when I create the equations and write them down, I always want to ask myself, what do the equations mean? Can I explain the result of these equations to a layman? And so that's what I really sought to do.
This you really can.
I want to do that.
So some of the things we've talked about over the last several years are much more complicated, relating to the vulnerable plaque, the interactions with elements in the blood, rapid changes in state and things like that. And I suspect that you're heading there over a period of time. I just wonder how you see this developing.
Well, yeah, the creation of this virtual plaque would be the first important step. I think the next step would be to get current model into arbitrary 2D geometries, right? So not just stick to concentric plaques, but to have general shaped plaques. So that would be the first milestone. And after that, you can start to think about incorporating different chemical reactions and different stiffnesses. That would, mathematically, all that involves is just putting more equations in. Actually, it's not conceptually, it's not difficult. Mathematically, it just means you put in more equations, and there'd probably be transport type equations for growth factors, for example. And these growth factors, they could change some of the material properties in that strain energy function that I was telling you about, right? The beta, the eta, those could be functions of the local growth factor density, right? And then you would be able to get changes in the material properties. You'd be able to simulate, for example, a part of the plaque getting stiffer or harder or more calcified because of the presence of certain diffusing species, for example. So that's where I see the next.
Then another material that you'll have to deal with over time is the necrotic core. So the necrotic core is soft,
right.
But I suspect doesn't you can't compress it very much. I suspect. I don't know, but I suspect you can't. So you have this sort of mush there that
right.
You move around.
So one thing I should say is that my current model is an incompressible model. Right. So that means that if you squish it this way, it's going to squirt out the other way. So it's incompressible already. So that part's already in there. Think about the necrotic core, as you said, it's pasty or it's mushy, right? Assuming that it can be modeled as a hyperelastic solid, right? That's a big assumption. Then you could potentially simulate that by saying, well, in the presence of say macrophages, right, which excrete these MMPs, then I'm going to locally lower the beta in my strain energy function, and that could mimic the creation of the necrotic core, because we're saying that you have normal intima here with a certain beta, right, certain material parameter, but when you add macrophages to this, then it becomes softer, right? And mathematically that means just lowering the beta parameter in the strain energy function, right? So these very complicated ideas to do with the medical side, they become quite simple if you look at them mathematically. But of course, there are assumptions. Right? We're assuming that the the solid is a hyperelastic solid, right? Some people might say actually the necrotic core is not a solid at all. Maybe it's more like a liquid, right? In that case, you'd have to refine the theory to account for that. Does that answer some of your questions?
It does exactly.
Okay.
But then on top of that, then you have these discontinuous functions of what happens with erosion or rupture of the intima, exposure of tissue factor and interactions with the blood. So then it's much more.
Right. It becomes very complicated, so that's why it's it's work in progress. The coupling with the fluid dynamics is obviously something that a lot of people explore. But the simulation of hemodynamics and blood flow in general is a very mature field in engineering and mathematics. So I don't really want to go there because it's already very, very mature. I think it's a lot better to develop the solid side of the plaque, to investigate the plaque by itself, and then later on engage in a collaboration with somebody who does fluid mechanics and hemodynamics, and then bring our two ideas together. So that's probably the best way to go about that. Hemodynamics is a very mature field.
What about relating this to sort of the more epidemiological, risk factor kind of models that that we developed? Is there are those things that can be plugged into these models? For example, I would think that one, as far as stiffness is concerned, I would guess age would play a role in that.
Right. I mean, in the model, because you get growth, you're going to get an increase in stiffness anyway, right? So that's going to be there anyway. But certainly, you could have material properties be functions of time. You could have the material properties. You could just say, I want the material properties to become stiffer as the patient ages. You could just put that into the model. Remember, in all that I've said today, I've been thinking about a single blood vessel, right? So one thing you could think about, and maybe this addresses your question, is kind of population level studies. So each one of us, we've got different plaque burdens, we've got different shaped blood vessels. So you could imagine initializing my model with by drawing the dimensions of the lumen, media, adventitia from a distribution, right? So you initialize the model randomly, corresponding to each of us in this room, and then you evolve it. You see how the population evolves, right? How does the population level intima thickness evolve with time? So the model could connect with population level studies in that way, and more epidemiological studies.
Maybe because from an epi perspective, one of the things that you look at is what's called modifiable risk factors. So you want to look at something that you can do, externally, like, for example, say diet or, whatever, something like that would, change, the dynamics on an individual.
I feel interested, I think, here is hypertension, treatment of hypertension.
Yeah, that is interesting. I mean, that is...
Hypertension presumably is going to may result in the arteries being stiffer. So you start out mechanical disadvantage, hypertension.
Right. This model, I could see it having connections with the hypertension theory, definitely. With hypertension, you get a lot of remodeling. Obviously, you get changes in blood pressure, right? That could be certainly an enhancement of this model, right? And I mean, in my current model, the blood pressure is actually inputted as a parameter. So I say the blood pressure is 100 milligrams of mercury, and that's it. It's fixed for all time. So obviously, that's a weakness of the model, right? But one thing that I'm quite interested in is trying to have the blood pressure as a response to the geometry of the vessel, right? Rather than having the blood pressure being a fixed constant in the model, having it be a variable that responds to changes in vessel shape, which is, I think, a bit more realistic. Yeah.
I enjoyed your talk a lot.
Thank you.
You've talked about, as you said, a single vessel model, basically. And one of the many fascinating things we have observed for a long time but remains unexplained about atherosclerosis in human coronary distributions is that there's not a uniform distribution of lesion development. Lesions are predisposed to develop in the proximal third of the coronary tree, and they tend to be more prevalent in certain locations.
Right.
Now, there's a tremendous amount of variability in what I just said.
Right.
But by and large, you can count on that bifurcation points and so forth, certain specific areas have a higher predilection for developing these lesions.
Right.
I think the traditional assumption has been that these points, for some reason, are just more sensitive to the triggering events for atherosclerosis or the accelerator events. But your model makes me wonder if there's something inherently different mechanically about the different parts of the coronary tree that might contribute to that.
Yeah, that is interesting. I never thought about. I mean, I was always aware of that, of what you said, that observation that bifurcation points, for example, they develop atherosclerosis more readily. But I never thought about that in the mechanics.
So a bifurcation point will have at least one area that's anchored and can't respond mechanically the way a cylindrical segment could. How does that impact your mathematical model?
So if it cannot expand, if there's going to be growth, but you're constricting the vessel, then the stresses will build up rapidly. My model will probably say that, right? I haven't exactly run that case, but if you're constraining the artery and you're not allowing it to move, but you're saying it's got to grow at the same time, then stresses will rapidly develop, and maybe those stresses kick off the disease. It could be that. The explanation that I've heard, which is I think a bit more common, is that the reason why the bifurcation points develop disease more readily is basically due to the hemodynamics. I don't know whether you've heard that explanation or not before, but at the bifurcation points, so the flow recirculates and you have these eddies. Right? The flow is not laminar, so it recirculates, and that results in low shear stress. And that's the low shear stress, low endothelial shear stress hypothesis.
Yeah. But the problem with the low shear stress hypothesis is that's about as far as it goes. It doesn't,
Right.
You say, okay, you have low shear stress. Well, why is that bad?
Right. So maybe that's one area, again, where mathematical models might help.
Yeah.
Right. So the hypothesis is low shear stress equals disease. But how exactly does low shear stress cause the disease?
Which if you just talk ask the average person walking down the street who was a mathematician or a clinician who didn't know about atherosclerosis per se, which is more likely to cause problems? Low shear stress or high shear stress? You'd say high shear stress.
High shear stress, right.
So why should low stress? Well, but you do have these eddies. But why should And I think that what Dr. Garrett and I've been taught is that somehow you're turning on cellular signaling.
Right. So they're saying that if you have eddies,
we don't really know.
If you have eddies, then somehow that influences the endothelium in a certain way, makes them more leaky, more permeable to lipids.
Something like that.
And then so
that stuff is not connected. It's just not, as far as I know.
Yeah. I mean that it would be nice if there was a simple explanation for that, in the same way that there's a simple explanation for the GR.
But there might be. I mean,
more work to be done. These are all very interesting questions. It's a lifetime of work.
It is. Other questions? That's fabulous.
Thank you.